So now we have RNA that's been transcribed from DNA, and we'll look at the second part of gene expression, which is translation of that RNA into protein. So we're going to take RNA, we're going to take messenger RNA, and now go through and look at how that RNA is arranged in codons to give us proteins. And then we're going to look at the roles of transfer RNA and ribosomal RNA, also involved in the process of translation. So translation is that second stage of gene expression. The first stage is transcription from DNA to RNA, and translation is that second part, taking that RNA that's now been transcribed and translating it into a final product of protein. In translation, we have messenger RNA that's needed. Messenger RNA is that code, that DNA code that has been taken and transcribed into a different type of code that can be read and understood by the ribosomes in order to make proteins. So messenger RNA is that go between DNA and the ribosome. It's going to actually bring that message from the nucleus out to the ribosomes. Ribosomal RNA, remember, is part of the ribosome itself. The ribosome is that protein making machine, and the ribosomal RNA is incorporated in that protein. The transfer RNA is RNA that's going to deliver the amino acids to the ribosome one by one. And so the transfer RNA is responsible for linking those amino acids together, actually forming the peptide bonds between the amino acids. Remember, in ribosomes, there is a large subunit as well as a small subunit, and these subunits come together to make the full ribosome. The messenger RNA, as you see here in yellow, the messenger RNA is read by the ribosome in 3 nucleotides at a time. So it's read in codons to determine which amino acid from the transfer RNA is going to appear next in that growing protein chain. And so now we're going to look at the translation in more detail to see how this works. All of the elements that required for protein are going to come together on the ribosome. Remember, the ribosome is protein and the ribosomal RNA. And so that is one of the ribosomes, is their role is to become part of that ribosome. The other components that are necessary for the translation of RNA into proteins is the messenger RNA, the transfer RNA, and the amino acids. Translation is going to occur in 5 stages. The first stage is initiation, second is elongation of that protein, termination of that protein. Then after these first 3 steps, when that protein has been fully translated, then that protein is going to, that protein inside of the rough endoplasmic reticulum is going to undergo folding and then processing in the Golgi apparatus. So 5 stages of translation in proteins. Initiation. Initiation is where the ribosome binds to the messenger RNA. So there's a region on the messenger RNA where the ribosome is going to bind, and then it's going to scan that sequence until it reaches a set of 3 nucleotides on the messenger RNA that consists of an A, a U, and a G. So once it gets to this codon, these 3 nucleotides, adenine, uracil, and guanine, that's where translation is actually going to begin. And so this codon, remember, codons are 3 nucleotides of RNA. 3 nucleotides of RNA equals 1 codon. Every codon is going to code for 1 amino acid. So this codon, adenine, uracil, and guanine, codes for the amino acid methionine. And so that first transfer RNA that's going to enter the ribosome, bringing that first amino acid, is going to carry a methionine amino acid. So the first transfer RNA with the amino acid methionine is going to enter the ribosome, and there has to be some type of a double check, a checkpoint, to make sure that messenger RNA is dropping off that amino acid in the right order. And so on the messenger RNA, you have your adenine, uracil, and guanine. And then on the transfer RNA that's bringing that methionine that this codon codes for, there is 3 other nucleotides that are going to serve as a check for the amino acid corresponding to the codon. And so these 3 codons on the transfer RNA are going to be complementary to the codon on the messenger RNA. So you see here, the A is complementary to the U, the uracil is complementary to the adenine, and the guanine is complementary to the cytosine. And so what these 3 nucleotides are called that are situated at the ends of the transfer RNA, these 3 nucleotides are referred to as the anticodon. So the first transfer RNA is always an AUG, which corresponds to methionine. It's a start codon. The first transfer RNA that is delivering that methionine to the ribosome is always going to have a check with the anticodon being UAC, which is complementary to the AUG. And so when the ribosome scans that sequence and reaches that AUG, or that start codon, the transfer RNA is going to bring the methionine that this AUG codes for, and the transfer RNA is going to bind to this AUG, as you see here, as a double check for that methionine that corresponds to the AUG. If by chance the transfer RNA that enters the ribosome is the incorrect, it has the incorrect amino acid, then the transfer RNA will leave out of the ribosome, taking that amino acid along with it. So this anticodon interacting with the codon is a double check to make sure no mutations are made. The next step of translation is elongation. So after the starting amino acid has been delivered to the ribosome via the transfer RNA, the next codon, for example, here is CUG. This next codon codes for leucine. Now, these are just codons that are being used as examples. This next codon can be any of those 20 amino acids that were listed in that master genetic code table. But in this case, the next codon is CUG, which stands for leucine. The second transfer RNA with the complementary anticodon, so now the second transfer RNA is carrying leucine amino acid, and the anticodon is going to be complementary to the codon to be that double check before it lays down that leucine in the ribosome. So you have the C on the codon is the complementary to the guanine, the uracil is complementary to the adenine, and the guanine is complementary to that cytosine on the anticodon. And so that first amino acid entered this place on the ribosome called the P site. The first transfer RNA always enters at the P site. All of the other transfer RNAs that come after that initial transfer RNA is going to enter the ribosome at the A site. And so that first transfer RNA entered the P site. The second and all the subsequent transfer RNAs will enter that ribosome at the A site. When there's two transfer RNAs in both of these sites in the ribosome, a peptide bond will form between the methionine and the leucine, or any other amino acids that are attached to those transfer RNAs. So these amino acids are going to form a peptide bond, and then this ribosome will shift so that now the first transfer RNA is in the E site, and the second transfer RNA then is in the P site. So now we see where the peptide bond has formed between the two amino acids. That second amino acid is still attached to that second transfer RNA, but now after that peptide bond has been made, the methionine is released from that first transfer RNA, and it has been transferred to the E site, and the second transfer RNA has been shifted to the P site. And so this entire little complex here of these two transfer RNAs have just shifted from the P and the A site over to the E and the P site. So you can think of the E site as the exit site, the P site as the peptide bond site, and the A as the acceptor site. So once this transfer RNA that has released its amino acid has been transferred to the acceptor, transferred to the exit site, it can now exit from the ribosome, go and pick up another methionine, and start another protein on another ribosome. The P site now contains that second transfer RNA with the growing chain of protein. The A site then is now open to accept the next transfer RNA that's going to correspond to the next codon on the messenger RNA. And so now you see that the first transfer RNA is leaving the ribosome. The second transfer RNA is holding on to that methionine and that second amino acid, the leucine. Then the third transfer RNA is entering the A site, carrying the next amino acid that corresponds to this codon. So this next codon stands for, it's got 3 nucleotides that stands for the amino acid threonine. Then the ribosome is going to shift so that this second transfer RNA will move over into the exit site, and this third transfer RNA will move into the P site. Then the leucine and the threonine will make a peptide bond. The leucine will be released from the second transfer RNA, and it will be free to leave the ribosome. And then the methionine, the leucine, and the threonine will be attached to the third transfer RNA in the P site. This process continues along the entire stretch of the messenger RNA until the ribosome reaches one of these 3 codons, UAG, UAA, or UGA. All 3 of these codons do not code for an amino acid. It codes for what's called a stop codon. And so once one of these codons is reached, that is the end of the protein. The messenger RNAs are released, the transfer RNAs are released, and the growing protein chain is complete and released. And then it can travel to the rough endoplasmic reticulum in order to be folded into the secondary structure. So let's take a quick look at the master genetic code. So as I mentioned, mRNA contains the codons. mRNA contains the code for those amino acids. And this code is universal among organisms, meaning that you can use this code to decipher the amino acids for all organisms. So we can look at the messenger RNA of all those organisms and determine which amino acid will come next in the growing chain of proteins. This code is also redundant. So it's universal in that all organisms can use this master genetic code to determine their amino acids, and this code is also redundant in that some of these codes, so you see, for example, leucine. Leucine has 1, 2, 3, 4, 5, 6 different codons that code for leucine. Proline has several codons that code for the same amino acid. And if you look at some of these other amino acids, you'll find the same thing. And so the code is redundant in that each amino acid, except for methionine, has several codons that will code for that same amino acid. And the reason being is that if there's a mutation, say for example in leucine, in this set of codons, if there is a mutation in this last codon, you can see whether it changes to G, to A, U, or C, it will still be leucine and it will still fold properly. So sometimes when there are genetic changes that occur in the DNA, that change can cause a change in the amino acid that's going to be next in that growing chain of protein. But if that mutation has changed in a way such that you still have the same amino acid, then you have what's called a silent mutation, a mutation that's not going to change that protein. Proteins change and become non-functional when there's mutations that result in a change of an amino acid. And so that protein sometimes is not folded the same way, or that change in the nucleotide can sometimes result in a stop codon being introduced before that protein is actually finished being translated. And so those proteins can, those mutations that occur in DNA can result in non-functioning or improperly folded proteins. So the code is universal in that all organisms can use this table to decipher the amino acids from the messenger RNA code, and it's also redundant in that more than one codon codes for a single amino acid. So in the next class, we'll review DNA replication, we'll review gene expression, and then we'll have a few examples of how we can go from DNA, transcribe it into RNA, and then translate that RNA into proteins using the master genetic code. We'll also have a brief lecture on gene regulation and how genes are regulated. Genes are not always transcribed and translated unless their products, the protein products, are needed inside the cell. And that allows the cell to save energy and use those resources of those building blocks to make things that are absolutely necessary for the cell. At this point, please answer the questions that you'll find on Moodle. There's a short quiz. You can answer those few multiple choice questions, and we will discuss these topics of DNA replication and gene expression in the next class.